of the recent research in my laboratory, I'm going to first focus on some of the physiological mechanisms underlying adaptive evolution during habitat change. And then I'm going to be talking about molecular evolution of some of the candidate genes and the population and genomic signatures of selection. So as you all know, invasive species are really bad. They cause destructive effects to impacts on the environment, and they cost billions of dollars per year in damage in the US alone. But one thing that's really remarkable about a lot of the invasions into freshwater habitats is that they disproportionately come from very, very different habitats. For instance, if we look at the North American Great Lakes, between 1985 and 2000, 70% of the invaders actually originated from brackish water habitats. In the Great Lakes, for instance, most of the invaders originated from the brackish waters of the Black and Caspian Sea region. Now this pattern is quite remarkable because actually an intermediate salinity of about 5 PSU tends to separate saltwater and freshwater species, such that there's a formidable biogeographic boundary that separates the geographic ranges of saltwater and freshwater species. So in light of that, the question is what accounts for the ability of some populations to invade novel habitats, when most of them in fact cannot. So to address this question, my lab has been using the charismatic, gorgeous copepod Eurythema apnis as a model system to address this question. So if you look at the native range of this species, it spans coastal waters in salt marshes and estuaries in North America, Europe, and in Asia. However, within the past 60 years or so, it's invaded many freshwater habitats throughout the world. So the question is, where did those freshwater populations come from? Well, our phylogeographic studies have found that the freshwater population shown here in white originated multiple times independently from genetically distinct clades in different geographic regions throughout the world. For instance, there have been multiple independent invasions in the Atlantic clade, shown here in red, and multiple independent invasions in the Gulf clade, shown here in green. So for a minute, if we zoom in on the invasions in North America, the saline St. Lawrence population invaded the North American Great Lakes about approximately 50 years ago. And if you look at the native range in the saltwater habitats, the ancestral saline population shows low tolerance under low salinity conditions and high tolerance under high salinity conditions. And under common garden conditions, the freshwater population, which is only about 50 years old, shows the opposite pattern of high tolerance under freshwater conditions and low tolerance under high salinity conditions. So given this dramatic change in physiological tolerance and also performance in other studies, the question is which particular traits are undergoing natural selection, allowing this dramatic shift to occur, of the freshwater population showing a dramatic increase in freshwater tolerance under freshwater conditions and a decline in higher salinities. So a great challenge for a small, cute little copepod like Eurythema apnis is as environmental salinity declines all the way to zero, it needs to maintain an elevated hemolymph or blood osmotic concentration relative to the environment. And this is very challenging against a steep concentration gradient, excuse me, and there are high requirements for ion uptake. So the question then is, how are ions taken up from a diluted environment? The mechanisms of this are actually really, really poorly understood. If you look at models of ion uptake from freshwater environments, the field is incredibly unresolved and highly controversial. And there have been different models proposed. For example, in Model 1, so taking up ions from a freshwater environment, I want to emphasize is really challenging. So in Model 1, the proton pump, D-type ATPase, would export protons out into the environment. And then using this concentration gradient, some other ion transporter, such as perhaps the sodium hydrogen antiporter, would then transport cations into the cell. Now, the identity of these secondary transporters that would cooperate with the proton pump is not well understood at all, but it's hypothesized that one of these could be the sodium hydrogen antiporter. In contrast to that, in an alternative model, ammonia transporter would transport out ammonia, and ammonia would then react with protons, creating ammonium ion, and this consumption of protons would then cause the sodium hydrogen exchanger to transport out protons in exchange for sodium. Now, I want to emphasize that the mechanisms of ion uptake from dilute environments is incredibly poorly understood, even for the human kidney. And there have been papers, really recent papers, talking about the role of this proton pump in human kidney in 2013 in PNAS. That's how recent this field is. And this Model 1 was developed pretty recently. The proton pump B-type ATPase was only known to be involved in ion uptake since 1991, and the sodium hydrogen antiporter was only first discovered in metazoans in the mosquito larvae Anopheles gambiae in 2007. So this is pretty recent. This model was developed in 2009 in fish gills. So the question then is, which model might be correct, or which model might predominate under different conditions, and might any of these ion transporters' functions evolve during saline to freshwater invasions? And so to address these, to test these models, and also to determine the loci that could be under selection during saline to freshwater invasions, we performed comparative gene expression analysis using illuminacy, conducting pairwise comparisons of expression between saltwater and freshwater populations. And we were interested in the shared expression between the multiple comparisons. And if we focus just on the genes involved in Model 1, they showed an increase in regulation or increase in expression in the freshwater population under freshwater conditions. And in sharp contrast to that, the genes involved in Model 2 actually showed a downregulation in the freshwater population under freshwater conditions. Now, to superimpose those patterns of expression onto the models, you can see here in Model 1, carbonic anhydrase, which actually supplies protons to the proton pump, showed an increased expression in the freshwater populations. And then B-type ATPase, the proton pump, which exports proton out of the cell, was also upregulated, as well as the putative secondary transporter, sodium hydrogen antiporter, which was first discovered in metazoans in 2007, was also upregulated. And in contrast to that, the genes involved in Model 2 were downregulated in the freshwater population under freshwater conditions. Now, this is from a common garden experiment, so these represent evolutionary shifts in expression between the saltwater and freshwater populations. So the fact that the genes in Model 1 showed an evolutionary shift toward increased expression in the freshwater population under freshwater conditions suggests that these genes were involved, the functional evolution of these genes contributed to freshwater adaptation. Now, I'm not going to belabor this point, but this is a slide I showed at a previous evolution meeting, but I just want to emphasize that the changes in expression do correspond to the actual changes in function of these transporters. For instance, B-type ATPase does show an increase in activity of the actual enzyme under freshwater conditions in the freshwater population, shown in light green, relative to the saltwater ancestors, showing that these actually do correspond to changes in function of the actual enzyme. So then we wanted to actually go into more detail and localize the sites of expression of these key transporters. So in order to do that, we performed in situ immunolocalization of these ion transporters. So if we look at B-type ATPase and sodium potassium ATPase, for instance, and they're co-localized here, they were localized at discrete sites of ion transport in organs that we named the chrysalis organs. So they were localized at the leg in these discrete sites that we named the chrysalis organs, which means salt leg. And interestingly, if we compare the freshwater and saltwater populations at these legs across the copepod, the freshwater population showed a significant increase in expression of B-type ATPase relative to their saltwater ancestor at legs 3 and 4. And this indicated that there was an increase in the number of enzymes per ion regulatory cell in the freshwater population relative to the saltwater population. And this increase in expression was consistent with the increase in gene expression and activity of B-type ATPase that I showed you in earlier slides. And interestingly, the sodium hydrogen antiporter that was newly discovered in metazoans in only 2007 in mosquito larvae also was localized at these sites of ion transport in the chrysalis organs. And this was a key ion transporter that was only hypothesized hypothesized to be cooperating with the proton pump B-type ATPase for ion uptake from freshwater habitats. So we explored the molecular evolution and evolutionary history of the sodium-hydrogen antiporter in more detail. And this is the first phylogeny of this ion transporter for metazoans. And one thing that's really striking about this pattern is for most organisms, there's only 2 copies of this ion transporter, such as Anopheles and Drosophila. Yet in the copod Eurythema avidus, we have a great expansion of this ion transporter of 8 copies and 7 tandem duplicates along the chromosome, right here. And that's pretty remarkable. And, one second. One thing that's really striking about this, the paralogs of NHA in the copod Eurythema avidus, is that the different paralogs have similar sequence, but they actually get reconstructed into very different protein structures due to especially an insertion. And that suggests that there are different functions among the different paralogs and copies of NHA in the copod Eurythema avidus. So we conducted studies of molecular evolution, and we explored signatures of positive selection in Eurythema avidus NHA paralogs, and found significant signatures of selection among the paralogs of Eurythema avidus, and between the copies of Eurythema avidus and those of other taxa. So then when we mapped these mutations on the molecular structure of NHA, we found mutations in the functional regions that correspond to possible changes in ion uptake stoichiometry. For example, this pH sensing region, which interacts with the ligand, and which mutations in this region would change the ion transport mechanisms of the ion transporter, had sites that were under selection in the copod Eurythema avidus. And likewise, this ligand binding region, which interacts with the ligand, also had sites that showed significant signatures of selection. So the fact that the evolutionary differences at the pH sensing region and catalytic sites had signatures of selection in Eurythema avidus suggested that mutations in these regions might correspond to evolutionary changes in function. So we found molecular evolutionary patterns across NHA paralogs and between NHA paralogs of different taxa. But then the question is, could we detect population genomic signatures of selection during invasions from saline into freshwater habitats at the population level? So in order to address that question, we performed a population genomics study where we conducted full genome sequencing for 100 individuals pooled per population for saline and freshwater populations in replicate across independent invasions in 3 genetically distinct clades. And we tested for population genomic signatures of selection for looking for distinguishing SNP allele frequency differences between populations and looking for FST outliers. And when we did that study, and this is an FST outlier analysis, and this line represents FST values that are more than core standard deviations from the mean, we see the ion transporters that show significant signatures of selection between the saltwater ancestral and freshwater invading populations. So in summary, we found parallel evolution, gene expression, and activity of ion transporters, and we were the first to find evolution of expression and function of ion transporters, B-type ATPase, sodium hydrogen antiporter, sodium potassium ATPase, and carbonic anhydrase associated with habitat invasions. We localized this evolutionary shift to the copod legs in new organs that we named the chrysalis organs, and we were the first to uncover evolution of B-type ATPase function associated with habitat change. We found signatures of selection in NHA genes, newly discovered in metazoans, and we found population genomic signatures of selection in this transporter and other transporters associated with saline to freshwater invasions. So what is remarkable here is the high speed to which these evolutionary shifts could occur, only 50 years in the wild and 12 generations in selection experiments in the laboratory. And the mechanisms here might be relevant for other invaders. And I'd like to advertise that my students will be speaking more on this topic. Nick Nathers will be speaking on Monday at 3 p.m. more on the molecular evolution of NHA, the novel metazoan ion transporter. Tiago is presenting a poster, Tiago's over there, on the population genomics of invasions. Martin will be speaking today on the copod microbiome during independent invasions. And Alex Meckler-Hickson will be talking about the cytochrome P450 superfamily, also today. Thank you very much.